Oh, I know what I forgot to do. Yeah, so this is been a crazy week because I spent most of Saturday getting stuff, getting bugs and critters for your aquarium and putting them in there. Anyway, but that killed a lot of time, which I needed to spend on grading things for you guys in my other class. So, I'm feeling like I'm running a little behind right now. Anyway, let's see if we can squeeze in some lecture here before the end of the day, right? All right, so anyway, locomotion. Last time we looked at cilia and flagella, which is used by many protozoans, as we saw in lab, but it's also actually used by a lot of smaller animals, which things like those acial flatworms you looked at last week. They basically glide on a coating of cilia that, I mean, it's too small to see under a dissecting scope, or you really have to kind of prepare them properly and slice them up under and look at them under a compound microscope. But they basically are coated in cilia, especially on the ventral surface, and that is what allows them to basically glide over the course of the surface. They don't really use muscle contraction very much. Although actually they can swim a little bit. I've actually seen them swim, but they're usually gliding along the substrate surface, and their muscles are mostly used just to kind of redirect which way their body is heading a little bit. So many small animals use cilia. Many larvae of larger animals use cilia as a major form of locomotion. But cilia but it's really kind of limited to relatively small animals for the most part. If you're larger, then you use muscles instead to provide locomotion. Muscles basically muscles can work by contracting, by forcefully shortening. Muscles do not push, so they pull basically, they contract. They can't forcibly extend. They extend by having things pulling on them and just not counteracting that tendency. So that means that muscles, first of all, have to attach to something, and they generally have to attach to something not necessarily strictly rigid, but at least relatively rigid. If they just act on soft tissue, the animal will just shorten, and if there are no counteracting muscles, then they just stay shortened. There's nothing that would cause them to really to stretch out. And so that means that if you have muscles, you generally need some sort of a skeleton. And the two major types of skeletons, in a broad sense, are hydrostatic skeletons and rigid skeletons. Hydrostatic skeletons basically use internal fluid pressure. They are less rigid and allow the animal to potentially change shape, while a rigid skeleton is generally, is, like the name says, is rigid, but it may have joints where it can flex at specific points, like hard skeletons do. So let's start with hydrostatic skeleton. Basically, a hydrostatic skeleton is where you have a fluid-filled compartment that is surrounded by muscles. And the muscle pressure then is basically applied on the fluid-filled container, and since water is relatively incompressible, that is, it doesn't compress much, if you squeeze on the container in one direction, it will bulge out in a different direction. So basically, typically most animals that use hydrostatic skeletons often have two sets of muscles. There are exceptions, like nematode worms, but most often have two sets of muscles. They have longitudinal muscles that run lengthwise, and so when those muscles contract, if they contract evenly around all of them around the organism, they basically shorten the animal, and as a result, because there's a fixed amount of fluid inside, and this is a closed container, the animal gets thicker instead, so the fluid causes it to bulge out. So you become, as the longitudinal muscles contract, you get shorter, you have decreased length, but increased width. So you're changing the shape using those muscles. If you contract longitudinal muscles on one side of the animal only, then only that side will get shorter, and the animal will bend. So that's what nematode worms only have longitudinal muscles. They lack circular muscles, which we'll get to in a second here. And so they basically flex on one side or the other to kind of bend and wiggle. All right, so if you so one thing you'll need to do though is if you don't want to get stuck this way, is you have to have muscles that work in a counteracting way if it's at all possible. And so many animals that have longitudinal muscles also have circular muscles as well. Circular muscles wrap around the animal, and when those muscles contract, they squeeze down, they make it narrower, they tighten a belt, and that means that that fluid gets pressed out in either direction. So when these muscles contract, so take this guy here, you contract these muscles, and what happens is that you extend the substrate, you extend the length, it gets narrower but longer. So you have two sets of muscles. One set of muscles makes the body narrower and longer. The other set basically makes the body shorter and broader. So you can have use these actions to basically change shape and allow the animal to work its way through the environment. Okay. Another trick that some animals use when you have a hydrostatic skeleton is that you can divide up that hydrostatic cavity, that fluid-filled cavity, into a number of isolated separate compartments, and so each compartment can potentially act separately. And so that's what the segmented worms do. It's a more sophisticated version. Rather than having a single fluid-filled compartment, you have multiple subdivided compartments. All right. So one of the key features and ways that hydrostatic skeletons work is that you can have these muscle contractions basically move along the organism in a coordinated wave pattern. So muscles can. So for example, here the lengthwise muscles at this end might contract first, while these are fairly relaxed and stretched out. Then these guys will at the front, the longitudinal muscles or lengthwise muscles relax, and the middle ones contract, the fat in the middle one, and so on. And so you have this wave of contractions, and you have this wave of bulge and narrow parts of the organism moving through the organism. And that's called peristalsis, is that basically a wave of muscle contractions, especially arranged in a sort of circular arrangement. And that can be used to do two things. You can move substances through a tube. So that's how your digestive tract works. For example, you push your food through your esophagus and through your intestines through peristalsis, through waves of muscle contraction that squeeze on your esophagus, and that squeezing just keeps working its way along the length, pushing the food ahead of it. But organisms can also use this to move through the substrate as well. So they can move food through a tube, or they can move themselves through a tube. They can burrow. So here's a diagram, a very schematic view of how this would work. So here we have a worm burrowing into mud. You can see that the anterior end of the worm right now is fairly broad because the circular muscles are relaxed and the lengthwise muscles are at the anterior end are contracted, shortening and causing the fluid to bulge out. Back here, the circular muscles are contracted so that they keep the back end narrow. Then what happens is these circular muscles begin to tighten, and the lengthwise muscles relax in the front, but that wave of contraction pushes backwards. So now you have the middle of the animal bulge out, while the front here pushes through the substrate, this bulge of muscle contraction here pushes through the back. This kind of acts to anchor it, while the front pushes through. Then you expand the front here again, you anchor the front while you draw the back into here, and you have these sort of pulses of anchoring and pushing and anchoring and pushing that allows you to basically move through a substrate. So and we'll see this in actual live action in about a month or so when we do the analytical lab. Looking you play with earthworms that move just like this through soil. And we'll have some live earthworms here. Okay. In contrast to hydrostatic skeletons, rigid skeletons are basically rigid, so they generally prevent gross changes in body shape, so they generally retain the shape of the body more precisely than a hydrostatic skeleton, where you can become short and fat or long and skinny relatively easily. So that means that with rigid skeletons, the organism generally has less flexibility in terms of the kind of shape it can take on. But it does have its advantages. It often allows for more precise movements because you're often restrained in the kind of movements you can make, but you can control those movements much more precisely. Root movements can often be much more rapid if you're dealing with muscles anchored to a skeleton, rather than having to act on a fluid that has to move through. So that response of the fluid is going to take time. So hydrostatic skeletons generally don't work terribly fast. In addition, there are other advantages beyond just acting for muscles to act on for a rigid skeleton as well. For larger organisms, they can provide physical support. They can hold you up. They can, in terrestrial environments, that may be particularly important to have a rigid skeleton that can hold you off the ground since you don't have the buoyancy of water. And a rigid skeleton can potentially, depending on how it's designed, but it can also sometimes provide protection against predators as well. Especially if it's an outer external skeleton, a shell or something like that. So rigid skeletons, in addition to providing faster and more precise movements, can also provide potential predator protection, and physical support as well, especially on land. So for rigid skeletons, we can further subdivide those. And by the way, even if an organism has a rigid skeleton, or skeletal elements, it doesn't necessarily mean the organism itself is rigid. It depends on how many parts there are to the skeleton and how they interconnect. But rigid skeletons can be subdivided then into endoskeletons versus exoskeletons. An endoskeleton is inside the body. It's underneath the skin at the very least. And it is usually derived from, it's usually of mesodermal origin. So it's derived from mesodermal tissue, embryonically speaking. Exoskeletons are outside of the body, so they're external to the skin. They're often secreted by the skin, by the epidermis, and since the outer skin is ectodermally derived, that means exoskeletons are generally going to be ectodermal as well, from ectodermal origin. And then just one other term that we've already seen in lab, but you should just be generally familiar with it, is the term test. A test is any hard, rigid skeleton, usually a single piece that's hard, that's rigid, and usually more or less external. There's a little squiggle here, because they're not always, strictly speaking, external. So, for example, you looked at the radiolarians in lab last week, or a week before, and they have a test. We call that a test, even though technically it's underneath the plasma membrane. So it would count as an endoskeleton, but it's close to the surface. And so it's almost external. A sea urchin shell, basically, is also referred to as a test as well. So it's a hard external covering, often taking the shape of the organism. We don't use the word test for things like clam shells, though, or snail shells for some reason. So both endoskeletons and exoskeletons are rigid, so that usually means that they have inorganic or mineralized components, but they also generally have organic components as well. So there's there may be proteins mixed in with that. Even if they're external, there's often organically produced substances within that skeleton. Our skeletons, for example, we humans have an endoskeleton. It is composed of calcium phosphate as a mineral component, but it's also fairly rich in protein, particularly collagen protein, and blood vessels and other things running through it. And in terms of how rigid skeletons originated, we don't really know, but there's some suggestion is that these mineralized skeletons may have actually risen evolutionarily as sort of an accidental byproduct of waste mineral production, and sort of sequestering that mineral, hanging onto it, and as it builds up from various metabolic processes, and then it turning out that that mineral actually is kind of useful. That is, can protect you from predators, for example. Makes you harder to eat. Or if you're in an environment that's maybe in fresh water, having a store up of minerals might be useful to draw upon when there isn't much in your environment. So it was originally thought that hard mineralized skeletons perhaps arose as a byproduct of sort of metabolic waste, of mineralized metabolic wastes, and from there kind of became a useful thing both for mineral storage and protection and things like that. And then after that, it evolved as a structural feature for muscles to act against. All right, if you have a rigid skeleton, your skeleton can be articulating. That just means it has joints, and therefore it has multiple parts that move relative to one another, like our own skeletons. Or it can be inarticulate, which basically means that it is a single rigid piece or a bunch of pieces fused together. So a snail shell, for example, would be an inarticulate skeleton, and the jointed legs of an arthropod are obviously articulating skeletons. A sea urchin tusk would be, because of those pieces that come derived from multiple pieces, but they're fused together into a single tusk, would be inarticulate. But the skeleton of a sea star, for example, is multiple pieces that move relative to one another, and so that would be an articulating skeleton. All right, moving on, a little bit more that we can say about skeletons. Skeletons, as I said, have a mineral as well as an organic component. There are various different types of skeletons, but the most common mineral in the majority of animals, and even some algae, like coralline algae, is calcite or calcium carbonate, CaCO3. There are various forms of calcite in terms of how it crystallizes and binds to water and things like that, but basically they're all calcite or calcium carbonate, and a few other carbonates as well. This is by and large the most common kind of rigid skeletal material. So it's the main component. It's what clam shells and snail shells are made up of, what sea urchin tusks and sea star skeletons are composed of, and so on. So most skeletons, most hard shells tend to be calcium carbonate in marine environments. Then less common is calcium phosphate, usually coupled with extensive amounts of collagen protein as well. That is what our bones are made up of. So our bones are not calcium carbonate, they're calcium phosphate. So they tend to differ a little bit. Otherwise, beyond vertebrates, however, calcium phosphate as a skeletal material is not very common. And I think the only other organisms that actually use it are some, not all, but some brachiopods. Brachiopods are shelled animals that look superficially like a bivalve mollusk. They have two shells, but only very superficially. And we'll cover them in 2 weeks, 3 weeks. We'll get to them pretty soon. But brachiopods, some brachiopods have a calcium phosphate shell. Other kinds of brachiopods have a calcium carbonate shell. Some things can have silica shells, not so common among animals. In fact, I'm trying to see if I can think of one off the top of my head. But it is found in some protists, like the radiolarians you saw already. And a few species can incorporate some silica into other skeletal elements. And then a third type is a strictly organic skeleton, which is a material called chitin. And this is a word that you need to make sure to spell correctly. C-H-I-T-I-N. The reason spelling counts here is because there's a set of creatures called chitons, C-H-I-T-O-N, which are a kind of mollusk, which we'll learn about later, but they're very different. So chitin is a fairly complex mix. It's a polysaccharide combined with several proteins. It's usually not as hard as a fully mineralized skeleton, but it's still fairly rigid. That's what the external skeleton of arthropods is made up of, and various other organisms also use chitin as well. It can be fairly soft and flexible if it's thin, but if it's thick enough, it can be fairly tough and reasonably rigid. And chitin can also be hardened with the addition of calcium carbonate. So things like crabs, which have pretty hard shells, get their hard shell because even though their exoskeleton is composed primarily of chitin, it also has calcium carbonate in it as well to make it even harder. So those are the basic components of skeletons that we'll find throughout the animal kingdom, with calcium carbonate really being kind of the biggest in terms of hard mineralized skeletons. All right. So with the skeleton done, let's move on to lunchtime food. All right. So when it comes to protozoans, and remember we haven't actually covered protozoans yet in lecture, but when it comes to protozoans, and just about all animals are heterotrophic. We basically define protozoans, which is as groups that are predominantly heterotrophic. If they were predominantly photosynthetic, we wouldn't consider them protozoans, we call them algae. And animals are essentially heterotrophic, although some use endosymbionts, the animals themselves are essentially heterotrophs. But remember, there's so much variation amongst the protozoans that there are lots of protozoans that can kind of be mixotrophic. They can be both. They can be heterotrophic and autotrophic, that is photosynthesized. So Euglena, which I mentioned here, but we didn't actually see in lab, but Euglenas and other similar protozoans, for example, often have chloroplasts, and so they are able to photosynthesize. The chloroplasts are where obtained from algae, and so they carry out photosynthesis and provide carbohydrates. But Euglena are also able to consume other organisms, other organic material as well. They feed on bacteria and small algae and so on. Dinoflagellates, another kind of protozoan, are basically a group that is ancestrally non-photosynthetic, but I think it's about half of all dinoflagellate species carry chloroplasts from brown algae and are photosynthetic and therefore photosynthetic as well. And in fact, some dinoflagellates have very complex life cycles where some stages are heterotrophic and some are photosynthetic, and it can be kind of quite a mess. All right, back to animals themselves. So animals themselves are heterotrophic, but it's not completely uncommon to have animals that rely on endosymbiotic algae, so not just the chloroplast, but the whole algae, and that are retained within their tissues. And those algae can photosynthesize and produce carbohydrates that the animal then uses either as a food supplement, as an energy source, or for some species it might be the primary source of energy, so that they would actually die if you kept them in the dark. The primary algae or protists that are used tend to be dinoflagellates. Photosynthetic dinoflagellates are the primary endosymbionts, but there are some other kinds of algae used as well. So groups of organisms that do this quite a bit are corals. Many corals, for example, not all, but many corals rely heavily on endosymbiotic algae. Some, in fact, probably rely almost exclusively for their energy on the endosymbionts and don't need to feed at all. Other giant clams rely heavily on endosymbiotic algae. Some anemones, some hydroids, you'll meet some of these in lab this week. Some sponges and some flatworms and sea slugs. So there's a number of animals throughout the animal kingdom that have evolved to be able to make use of endosymbiotic algae as a source of additional energy, or sometimes even as their primary source of energy. So the animals themselves, if you kill off the algae, the animals themselves would still be basically heterotrophic. They're relying on an autotroph living inside of them to provide that energy. All right. So, skipping past the photosynthetic guys there, both protozoans and animals need to obtain their nutrition in some way from other living things, basically, which means that they have to obtain generally large food particles. Since the ocean's not swimming in sugars and amino acids, you generally have to obtain larger food particles, entire cells and other organisms, and break them down via digestion. That digestion can take place either inside or outside of the cell. So extracellular digestion is where the large molecules are basically broken down outside of the cell, and then the individual small molecules, as they're released, the individual sugars and amino acids and things like that, are then transported directly across the cell membrane using either diffusion or active transport or something like that, typically involving specialized channels for transporting them. This kind of extracellular digestion is typical of fungi and fungus-like protists that grow in their food. They secrete digestive enzymes into the external environment, and then they absorb many of the nutrients that are released as a result of that. We humans basically also rely primarily on extracellular digestion. That we break down our food in our guts before it actually passes through any plasma membrane. All right. In contrast to extracellular digestion, intracellular digestion is where large molecules and food particles, including things like other cells and fragments of cells and so on, are taken into the cell and digested inside the cell, inside specialized vacuoles, digestive vacuoles essentially. This is how most of the critters we generally think of as protozoans basically do their digestion. It's an intracellular. That is, think of an amoeba. They use their pseudopods to extend around the food particle. Something like a paranema or paramecium will take food into their cytostome, their sort of mouth cavity, and basically pinch off a vacuole of food. So the digestion takes place inside. One of the main methods of taking up food is a process called phagocytosis. So phagocytosis basically involves, so here's a plasma membrane of some cell that's not even shown in its entirety. This is its membrane. Here's a food particle. So it will basically surround the food particle. It will either pinch inwards to trap the food particle, or it can even extend pseudopods outwards around the food particle. So one of the advantages for amoebas having pseudopods is that it can allow them to eat relatively big things. It can extend those pseudopods around the relatively large food particle and capture that food particle inside a vacuole. That vacuole will then combine with lysosomes, which are small little vesicles that basically contain digestive enzymes. And those digestive enzymes break down the food particle. It might be an algae cell or bacterial cell. And then as those substances are digested, they're released into the rest of the cytoplasm for the organism to use. All right. So when it comes to animals themselves, remember one of the things I said that sort of tends to define animals is that they're generally ingested. That is, their food processing takes place inside in some way. There are some exceptions, or some borderline cases, not exceptions, these borderline cases, but that's a generally true. Most animals have an internal digestive area, a digestive tract called the gut. Food enters into the digestive tract, into the gut. Digestion takes place in the gut, and then the nutrients are absorbed. And for most animals, there are exceptions we'll get into later, but in many cases, the food is I'm not sure I want to put a percentage on that. But certainly in our guts, for example, digestion is extracellular. The digestion takes place in our gut, but food that's in our gut hasn't crossed any plasma membrane yet. It's not within any kind of a cell, so it's still kind of, the gut is open to the external environment, and so that's still extracellular digestion, and then the gut absorbs the individual sugars and amino acids and so on, the small particles. In other organisms, food particles can be captured, even if from the gut, they can be captured via phagocytosis and then digested intracellularly. So both are possible. All right. If I had touched upon this very briefly already, but incomplete. Actually, we did talk about it at least in lab. If you have a gut, your gut can be either incomplete or complete. An incomplete gut or blind gut has only a single opening. So material enters into that opening, but then anything that's not digested has to exit through that same way. So there's only one opening. That means you're basically, vomiting out your waste products, I suppose. And that is found in, for example, the cnidarians, which is what we're going to look at in lab this week, and also things like flatworms, platyhelminthes, flatworms also have only a blind gut digestive tract as well, And it's actually secondarily involved in other animals as well occasionally. So brittle stars, for example, don't have an anus, they only have a single opening. In contrast, a more sophisticated type of gut is a complete gut, which has two openings, a separate mouth at one end and an anus at the other. What that means is that food enters in one direction, it enters through the mouth, gets processed, and it exits at the opposite end of the animal. Or not necessarily the opposite end of the animal, but the opposite end of the digestive tract, because sometimes it can loop back to the other mouth. But the basic point is that in a complete gut, food is going to flow in one direction. With an incomplete gut, anything that goes in, you have to then expel again. So that means that you have to completely process your food, get rid of any undigestible waste before you can eat again. While with a complete gut, you can process, you can have some food come in, and you can have and keep eating even as the others before you eliminate all of the other waste. You have a single directional flow of food, and so that makes that gut more efficient, that you can continue to keep eating. It also allows you, because you're basically working in a kind of assembly line fashion, you can also have regionally specialize your gut. So you can have the different part, first part of your gut can break down big particles, the second can be more for chemical breakdown, the third part can be absorption of nutrients. So you can have much more specialized regions of the digestive system to further increase the efficiency of the whole thing. And so again, that's something to look at when we look at the various organisms. What kind of a gut do they have? Incomplete or complete? All right. And so now let's go and look at a little bit about what organisms eat and their basic strategies. So the basic feeding strategies include herbivore, where you feed on plants or algae. So that's an herbivore as opposed to a carnivore, which feeds on other animals, and omnivore, which eats a variety of different plant and animal matter. One kind of common type of herbivore are grazers. Grazers are animals that feed on plant or algae tissue without consuming a whole organism, without consuming a whole plant or algae. We actually apply that term also to certain kinds of carnivores that might be feeding on sessile animals. So if you're feeding on something like corals, or feeding on bryozoans or sponges that are growing across the substrate and not moving themselves, you may be just taking little nibbles off of them, not killing them outright, and so that's often referred to as grazing as well. You're feeding in a similar way that an herbivore might feed on a plant. All right, so those are the basic sort of what you're eating definitions. And the next thing is how do you feed? How do you catch your food? One way to catch food is through what's called suspension feeding, sometimes called filter feeding, but that term is kind of vague and is kind of used a little bit imprecisely. So scientific parlance, we generally prefer the term suspension feeding. In suspension feeding, you trap and filter small particles from the surrounding water column. For example, this feather duster worm here has a bunch of tentacles. It lives in a tube. It has a bunch of tentacles with lots of fine little branches. As water currents flow past it, little food particles get stuck in those tentacles, and it draws them in towards its mouth. So it relies on a movement of water past the feeding structure in order to capture food. Typical suspension feeders are obviously eating small food particles. So they may be eating bacteria, phytoplankton, that is single-celled algae, zooplankton, small little animals and animal larvae and so on. Detritus, which is basically bits and pieces of dead organic material, little bits of seaweed or animal poop or whatever it might be, but generally very small particles that they capture with their tentacles and bring into their mouth. Something like a feather duster worm, which is not really capable, which is fairly sessile, it lives in a tube here, is pretty much stuck with relying on water currents to bring the food to it. So it basically sticks its tentacles out somewhere where there's flowing water in water currents, and the water currents are carrying food particles, and which they try to trap. If you don't, but you can also create your own water currents. So for example, you can move your feeding structures through the water. So up here you're looking at a giant acorn barnacle. Acorn barnacles are arthropods, so they're actually they're crustaceans. In fact, they're related to crabs and things like that. But as adults, they lie on their back. They secrete a bunch of calcareous plates, and what used to be their legs are now used as a filter feeding structure. So you can see they extend out these legs, which have lots of little bristles on them to catch food. But these guys are much more active than a feather duster worm in that they're constantly sweeping their legs. They're sweeping their legs through their environment, through the water, trying to catch food. So they're not necessarily relying on just water currents themselves. They're actually moving their feeding structure through the water. And so that's an acorn barnacle. The other possibility is to actually pump the water through your feeding structure. So here, for example, is a worm called a diagram of a worm called a parchment worm. Parchment worms live in tubes tube-like burrows in the sediment that are U-shaped. So the burrows open up to the water above two spots at the anterior and posterior end. The worm lives in here, and in this big space right here, it secretes a huge big bubble of mucus. That mucus serves as the filter feeding structure, as a suspension, that is small particles get stuck in the mucus, and then it just consumes its mucus wall and generates a new one. But it has these special appendages down here that basically pump water through the burrow. So they pump water in this way and out this way. So they create a steady flow of water through their burrow, which then brings in food particles that their mucus wall captures and they can eat. So they're pumping water through their feeding structure. These guys are moving in a feeding structure through the water. And something like a feather duster worm is basically just passive. It just sits there and lets the pre-existing water currents bring food to it. And so I think this will be the last slide we'll cover today. So the food particles can be trapped in various ways. As you saw with that worm, they can use mucus. You can have mucus-covered tentacles. So here, for example, is a tube anemone, a member of the Cnidaria, happens to live in a tube. It has a bunch of tentacles here. Those tentacles are covered with mucus, and food particles stick to the mucus and then are brought to the mouth. They can be moved through the mouth, often using cilia along there to move that mucus through the mouth. They can, or if you're eating somewhat larger particles, you might have tentacles or 2 feet or something. You might actually be able to grab the food particle that intersects you. Or you can have very finely branched bristles, as we saw with the feather duster worm. You also see that in the shrimp here, just to give you a little bit of taxonomic variety. So instead of having a normal little pincers to grab food, this shrimp here, you can see that it kind of looks a little blurry, but that's actually because the front legs have a very fan of very fine hairs. That's why it's called a fan shrimp. That basically act to trap food particles here, very fine bristles, or hairs along its legs here that spread out to form this fan-shaped thing. And it just waves those through the water, trying to trap small algae and other kinds of food particles like that. All right, so that's it for, we'll go back, we'll continue talking about food on